the next topic, there is a culture of the bacteria, which I said earlier. That what is culture media? I already explained in brief. So let's move to that. A nutrient material prepared for the growth of microorganism in a laboratory is called culture media. Okay. Some bacteria can grow well on just about any culture media. Other require special culture media, and still other cannot grow or any non-living medium yet develop. Okay, so few bacteria may grow on a specific type of culture media. Some bacteria can grow in any kind of culture media, and few microorganisms may not grow in artificial culture media. Okay, so microbes that are introduced into the culture medium to initiate growth are called an inoculum. Okay, the proportion of the microbes which you add in the culture media for the observation of the growth. Okay, this quantity of the microbes are regarded to be as the inoculum. The microbes that grow and multiply in or on a culture media are referred to as a culture. Okay, now ingredients of the culture media. What are the basic ingredients which are basically used as a component of the culture media? Agar or simply the agar. Okay, there is a component. Now, agar is the main component that is used universally for preparation of the solid media. Okay, a solid media, liquid media, this kind of medias are available in terms of the culture media. So, to form a solid media, we require agar or simply the agar. It is obtained from a variety of seaweeds, and after necessary processing is usually available as powder or as long threads. Okay, agar is chiefly composed of a long chain polysaccharide consisting of D-galactopyranose unit, a variety of inorganic salts, minute quantities of the protein-like materials, traces of the long chain fatty acids, and minerals such as the calcium and magnesium. These all are the ingredient of the agar. Next, peptone. Peptone is another important ingredient of culture media. It is a complex mixture of partially digested proteins. It is obtained by digestion of lean meat or other protein materials such as the heart muscles, casein, okay, fibrin or soya flour with proteolytic enzymes such as the pepsin, trypsin and papain. Okay. Other ingredients which are generally present in the culture media, other common ingredients of the media include water, sodium chloride and other electrolytes, meat extract, yeast extract, malt extract, blood and serum. Meat extract is available commercially as lab lemco and contains inorganic salts. There is a name, that is lab lemco of the commercial name of the meat extract, and contains the inorganic salt, carbohydrates, certain growth factors, and protein degradation products. Blood or serum is usually used for enriching culture of the bacteria. Okay, because blood and serum is a huge source of the protein, right? Usually, 5 to 10% defibrinated sheep or human blood is used. Okay. Now, chemical and physical types of the culture media that we need to talk at first. Simple media. Okay, one by one we will discuss about the various category of the culture media. The first topic, that is simple media. The simple media or the basal media include nutrient broth and peptone water, which form the basis of the other media. Broth means the liquid medium, right? Nutrient broth is an example of a simple liquid medium that consists of peptone, meat extract, sodium chloride and water. Addition of 0.5% glucose to nutrient broth makes it glucose broth. Okay. Nutrient agar is an example of a simple solid medium. The medium is used routinely for isolation of many bacteria from clinical specimens. Next topic is the defined or synthetic medium. A defined medium, a medium in which all chemical components are known is called defined or synthetic medium. Known means you have to add in required proportion. That means how much you need to require a particular component within a medium, that you have to decide by means of the characteristics of the inoculum. Okay, that's why the proper quantity, in the previous in the previous culture media, in case of the simple media, there we know these are the components which are present, but how much they are present you don't know. Okay, that means you know the quality, but you don't know the quantity. But in case of the defined or synthetic medium, you know the quality as well as the quantity because it is formed by you. Okay, it can be in a liquid form, that is broth, or solidified by agents such as agar. All the chemicals used are known and it does not contain any animal yeast or plant tissue. These media consist of trace elements and vitamins, a defined carbon source and nitrogen source required by certain microbes. Glucose or glycerol is often used as carbon sources and ammonium salts or nitrate as inorganic nitrogen sources. Okay, Dubose medium with twin 18. Dubose medium with twin 80 is an example of this medium. Okay, these are the examples. Complex media. Media that contain some ingredient of unknown chemical composition are called complex media. That means all the components which are present in that particular media is not known. Okay, in terms of the quality as well as the quantity. They are very useful because a single complex medium may be able to meet all the nutritional requirement of many different microorganisms. In addition, complex media often are needed because the nutritional requirement of a particular microorganism are unknown. You don't know previously. Suppose there is an unknown microbe, and how would you know that what kind of nutritional requirement is there for that particular microbe? You don't know because it is an unknown microbe. So if you culture that particular unknown microbe into the complex media, as because within the complex media we have all the different category of the nutrients, so that will be a suitable media for this unknown type of microbe. Okay, complex media are also used to culture microbe with complicated nutritional and cultural requirement. Cultural means by means of their isolation area. Okay, most complex media contain undefined components such as the peptone, meat extract, and yeast extract. Peptone are protein hydrolysates uh, prepared by partial proteolytic digestion of meat, casein, soya meal, gelatin, and other protein sources. They serve as a source of carbon, energy, and nitrogen. Beef extract and yeast extract are aqueous extract of lean beef and brewer's yeast, that is a type of yeast, respectively. Beef extract contains amino acid, peptide, nucleotide, organic acid, vitamins, and minerals. Yeast extract is an excellent source of um, vitamin B or vitamin B complex as well as nitrogen and carbon compounds. These commonly used complex media are nutrient broth, okay, typic soy broth, and McConkey agar. These are the examples. Okay, nutrient broth, triptych soy broth, or McConkey agars. Now, if we're going to talk about the functional types of the media, okay, the first is enrichment media. An enrichment medium contains special nutrients that allow growth of a particular organism that might not otherwise be present in sufficient number to allow it to be isolated and identified. Suppose a small quantity of microbes is present and if you just want to culture it in a large volume, so you have to select the enrichment media. Okay, where you have to put this small quantity of inoculum and by means of the enrichment media, by using the ingredient of it, they become they can grow, increase their population. Unlike selective medium, an enrichment medium does not suppress other. Okay, so any sort of unknown bacteria or a mixed culture of a bacteria, mixed population of a bacteria could be cultivated at the same time. Okay, selective media always select a specific type of microorganism. They will not allow the growth of another microorganism. Suppose you have a complex or mixture of microbes, and within that mixture there is suppose present of A type, B type, and C type of microbes. And if you put that complex into the, or if you just put that mixture of the microbes into the selective media, so any one out of these three microbes will be cultivated or will be survived and other two will be died. That's why there is selective media. In case of the enrichment media, if you put the same component, mixture of microbes into the enrichment media, then what will going to be happen? All the three can be grow proportionally. Okay, for example, because Salmonella typhi organisms may not be sufficiently numerous in a fecal sample, that means in a stool sample, to allow positive identification, They are cultured on a medium containing the trace elements selenium, which support growth of the organism. After incubation in the enrichment medium, okay, the greater number of the organism increase the likelihood of a positive identification. Okay, selective media. I already talked about the selective media. Selective medium is one of that encourages the growth of some organisms but suppresses the growth of the others. For example, to identify Clostridium botulinum in food samples suspected of being agents of food poisoning, the antibiotics sulfadiazine and polymyxin sulfate, okay, are added to anaerobic cultures of Clostridium species. Okay, this culture medium is called SPS agar. Okay, because of these two ingredients, it allows growth of the Clostridium botulinum while inhibiting growth of most other Clostridium species. Clear? Differential medium. A differential medium has a constituent that causes an observable change, a color change or a change in the pH in the medium when a particular biochemical reaction occurs. This change allows microbiologists to distinguish a certain type of colony of the microbes from other growing on the same plate. SPS agar also serves as a differential medium. Colonies of Clostridium botulinum formed on this medium are black because of hydrogen sulfide made by the organisms from the sulfur containing additives. Okay, so any kind of black spots over the culture that helps to microbiologists to isolate only the Clostridium botulinum. Okay, from a mixed culture by means of the differential media. So that's why it is the differential media by which you can differentiate. Okay, many media such as the SPS agar, McConkey agar are both selective and as well as the differential media. McConkey agar contains crystal violet and bile salts which inhibit growth of the gram-positive bacteria while allowing the growth of the gram-negative one. McConkey agar also contains the sugar lactose plus a pH indicator that turn colonies of lactose fermenters red and leaves colonies of non-fermenters colorless and translucent. Okay. Transport media. Transport media are used to maintain the viability of certain delicate organisms in clinical specimen during their transport to the laboratory. They typically contain only buffers and the salt. What? This particular media only contain buffers and the salt. They lack carbon, nitrogen and organic growth factors, hence do not facilitate microbial multiplication. Example of transport media are Stewart's transport media or Neisseria gonorrhea. Okay, we just need to prepare the transport media for the survival of the microbes, not for their further growth. Okay, after the isolation of a sample, we just need to put this kind of microbes in the transport media until and unless we examine the microbes. Okay, we no need to allow the growth of them. Okay, that's why it is called the transport media. Sugar media. Sugar media basically contain 1% sugar, which in microbiology denotes any fermentable substance as such as glucose, sucrose, lactose, and mannitol that is routinely used for the fermentation test. Okay. Now, how we can prepare the media? How we can prepare the media? Some naturally occurring substances are used for the cultivation of the bacteria. Notable among these is milk, such as natural materials are merely dispensed into the tubes or flask and sterilized before use. Okay, media of the nutrient broth or nutrient agar type are prepared by compounding the required individual ingredients, okay, or increasing the proportion of the required individual ingredients, or some conveniently by adding water to a dehydrated product which contain all the ingredient. Okay, that means suppose you have a powder, you just need to add the water along with the agar by means of which you can prepare the media. Okay, and if you just omit the agar, you can prepare a liquid medium that is regarded to as a broth culture. Okay, practically all media are available commercially in powdered form. The preparation of bacteriological media usually involve the following steps. Each ingredient of the complete dehydrated medium, that is powdered form, is dissolved in the appropriate volume of distilled water. The pH of the fluid medium is determined with a pH meter and adjusted if necessary. If a solid medium is desired, agar is sorry, a solid medium is if the solid medium is desired. Okay, so agar is added and medium is boiled to dissolve the agar. The medium is dispensed into tube or flask. Okay, there you have to hold it, or you can also dispense it into the or over the petri dish. Okay, the medium is sterilized generally by autoclaving. I already explained what is the meaning of autoclaving. Some media or specific ingredients that are heat labile are sterilized by filtration process. Okay, and that is all about the preparation of the culture media. So go for that particular video. In next video, you will come to know about the different growth aspect of the bacteria. Thank you.